Good
Just
eye.
got a little excited.
I'm zooming out all the way.
The ship is stopped already, right, Sam?
Sure
Like
is.
Yeah.
Hopefully that pans out. You see
Yeah.
it, you see it going too far one way or another. And then
What collection method are we thinking for the seawit?
This is going to be a floater for sure, so I would slurp it after you snip it.
okay, Al, can you zoom in?
Nice. Beautiful.
So fine.
Yeah.
Okay. Got those captures. Can we get a laser shot of the whole thing?
Yeah, turning
You want to
lasers
look
on.
at the base?
No.
No.
Sure.
I'd have to move.
You want to get the full wide shot first with lasers? Before we move?
That's fine. Yeah, right there is good.
Wait, just a touch.
All right, all set.
Zooming out.
So snip and slurp.
It's a snip and slurp.
How much are we snipping?
So, I'm thinking like maybe here-ish. So, so that's what, maybe one complete rotation, just to be safe.
Yeah, that sounds good.
Pretty ropey.
It will snap. There it is.
Just had to say so.
I think this is jar number 1. It's hard to read with the glare, but
Yeah, it looks one to me.
do you want me to pan down?
Sure.
All right, ready for section?
Yep.
There's nothing in jar 1, right?
Nope.
Yeah. Other than apparently a dirt cloud that's going to make it hard
That was
to see.
probably sediment in the tube. I think I see it.
Something in there.
That wasn't it.
Yeah, that looks like a tube worm.
Bonus.
I don't think that was it. I think you need to keep sucking.
If you turn off the porch light Would we have better light on that
Maybe.
jar?
Porch light's off.
No. Well,
that camera has its own light.
oh, okay.
Do we see it in there?
Turn it off.
Yeah, turn it off, Paul. Ooh, that thing's unhappy.
Ooh. Where'd that come from?
Wow. That's Dr. Seuss like.
Yeah.
That is a weird worm.
It's very unhappy.
Oh.
Now he's happy. So do we need, I guess we need to shake the hose some.
And run the jar?
Yeah.
Steve, is this 061? Or will it be
That's right.
061?
Yeah.
Copy that.
Turn it off for a second.
Yep, there it is.
Yay.
Nicely done.
Mm. Lizzie, do we want to pick up a sea pig and take a call here? Or do you want to keep moving?
I don't see why not.
Worked out.
Let me take another look at
Wow.
Good math.
iPack.
Okay, I'm going to do a DVL reset here.
Okay.
I'm thinking,
Well, I mean, judging by,
yeah.
we're out in front of Argus by probably 10 something meters.
That doesn't make any sense.
I mean, Argus is out in front of us. Yeah.
So
I don't think you should rely on this very much at all.
no.
Go by that reality.
Yeah.
So I know that's way off the cloud, but the likelihood is, you know, he came out this way,
Yeah.
so that you'd get, you know, Argus would be behind it a ways.
Back here somewhere. Somewhere
Well,
up
I mean,
here.
it's coming off of the stern, right?
Yeah.
So it's probably, yeah,
That one.
It's come out here. Argus is probably there, reality, but I mean, then you offset everything, you know?
Yeah.
I don't know if that's worthwhile. I think it, yeah, then Argus jumps around so much. I don't know.
Well,
I don't know if it's
worth doing.
worth doing. Yeah,
Okay. As long as we both agree that it's not correct.
not correct.
Roger, not correct.
We're too deep for that system to be
Okay.
usables.
What are we thinking, science?
So I'm just trying to assess in terms of the total length of time that we have on the bottom. Lisa was estimating that to be 18 hours. And it looks like we've come, or we're able to transit probably a quarter of all of the points
Are we looking
since
for
the
a
beginning of
CPIG?
the dive.
We want to find that while we're talking?
Science, are you interested in a CPIG here while we're trying to figure out what's next, since we're going to have about 10 plus minutes to put in a ship
Yeah.
move? There's a
There's one
CPIG right
there.
there.
There's one right there.
Yeah, and let's, well, the last, when we were collecting for Taylor, she wanted several C-pins, so I'm wondering if she's going to want several C-pigs. Steve, do you recall if she collected multiple?
She collected several C-pens from one small patch in a slurp.
Yeah.
I don't know if she needs
Mhm.
several C-pigs, maybe just one or two.
Shoot.
How big's this
Okay.
guy?
She's coming on in half an hour. I don't think they'll disappear in
Oh.
half an hour.
Okay.
We just keep moving on and then worry about sea pigs at a later date.
Because how many do we see in like this immediate area? I see the one that's right in front of us. And I thought there was another one off to the right.
Yeah, there's one off
maybe?
to the left. That's one over there, right?
Yeah, looks like
Yeah.
it.
So there's at least two.
Okay. Because I would just want to make sure that we got her everything we needed. So maybe multiple sea pigs and 2 sediment cores.
When I see two, I don't see, without looking around some more, I don't see more, but
Sure.
probably more.
Oh, wait. And we said that was not a slurp?
Well, it's got to be a
Small sea pig.
Yeah, I don't know. Can you zoom in on? It's fairly wee.
I'm sorry.
That's one way to say it's small.
Yeah. Hey, wee lad.
I don't know.
Oh. Yeah, why don't we take this sea pig and some sediment cores
Okay.
and say that we gave it the good old college try.
Coming out, Bob.
Because if we wait a half hour, we're going to have to stop the ship again. And we're already stopped.
That's true.
Here, why don't you get the sea pig?
All right, we've got Paul on the Herc arm for sea pig collection.
Oh, what jar are we going to do this in?
You can go to anyone but one.
Anyone but one.
Steve, are we close enough to keep this as the same target as the sea whip, or you want a different
I
one?
think it's fine to keep it the same.
Roger.
Sorry, Mr. Sea Pig.
That's right.
Can we zoom in at all, or like get the camera on there at all? Is that already?
Oh, you got that much.
You got jar 2 queued up there?
Could just go with a hose and not bother with that handle thing.
Could just what?
Just use the hose and don't bother with the handle.
Oh.
Might be harder, but or you could drag it out so you can get to the handle easier.
Yeah.
For suction?
Yeah.
You know, that seems
Oh, there it goes.
going in.
Oh, no. Oh, no.
That's what we wanted.
That didn't even seem right.
I think it was
No.
small enough for the hose.
Come on, sea pig. Hey,
There
it
it is.
is. Yay, score.
Nice. Perfect size sea pig.
All right.
Can we leave that camera on that jar so we can see
Yep.
what it does?
Yeah.
See if it rights itself.
Like a cat.
He's a lot more rigid than I anticipated. Oh.
After that trauma, I can
So,
imagine he's pretty uptight.
So tube cores
Yeah.
now, or what's the scene?
Yeah. The two cores are kind of in the area where you collect the C-pens or the whatever filter or deposit Peter she's collecting, so she can get an idea of, in the same area, the microplastics to see if they're consuming them.
You can always look on the screen and it'll tell you
Mm.
if it's locked or it's indexed.
It says press index button to yeah, got it.
All right. Bob's got arm back for this one.
Ready for tryout?
Ready.
I'm just gonna go halfway because we have that floaty one in the back.
Yeah. That's probably good right there.
All right.
Actually. Hopefully.
Can we zoom in on Al? Can we zoom in on Argus too?
You switched that other camera over too?
Oh yeah.
Not that I need it this minute. I will. So in the area of the pig area?
Oh, no, anywhere.
Good? Like it?
Yeah.
Looks pretty good.
Should we do the Bob Waters method? Put two in and then pull them out?
That is a better way to do it. Stir up the bottom. Ugh. That's not my method anyway.
It is now. If you're doing it,
I'm not.
it's your method.
I didn't come up with that method. Sort of standard. Ain't gonna work.
That's so cool to watch in the Argus cam.
Oh, that's a great angle.
Oh yeah, screen grabs.
Any old place?
That looks good.
Any old place will do.
Coring in action.
Nice. For anyone watching, we're currently taking sediment core samples in the soft bank area off Santa Lucia Bank in Southern California. We're partnering with a student who is looking at microplastics in the deep sea for her master's thesis. And so she's collecting some filter feeders and some deposit feeders from the deep sea. And she's going to analyze their gut contents to see or characterize if there are any microplastics in their gut system. So she's also sampling the environment to have a better idea of the context of if she's finding a lot of microplastics, are there a lot of microplastics in the environment? Or vice versa, are there not a lot of microplastics in the sea cucumbers, but there is in the environment? So are they selecting, are they ingesting,
It's got to come out.
are they retaining?
Oh, okay.
Yeah, I get hung up on the phone right there. Maybe.
I can come out more.
I think I'm okay. Great.
And I would say if there is another sea pig that's kind of close by where we don't have to ask the ship to move, I say that
Yep.
we should probably collect that one, just one more.
Not too far. You can get the next pig, too.
Let's come light on Argus.
There's been a Kirk arm handoff again. And time check, we've got it's 11:42.
Thank you.
So I think there's one right over here, right? Oh, no. Dang it.
If you need it to be small, you can also keep an eye out for
Yeah.
the smaller one.
Must have heard we were looking for it.
That one?
Right there
Where?
on the right. Maybe that's an urchin. Up here.
I think we just passed over one.
Really?
I thought we did. Maybe everything looks like a sea pig at this late at night.
Yeah, it's pretty messy right now. I gotta only go forward.
Okay.
Oh, there's one there, right? That one?
Yep.
Yeah. Is it? No. Yep. Yep. Nope.
Yep.
Yep.
Beep, beep, beep.
Okay. Oof,
Small enough?
that pig pen.
Looks like it has longer appendages than the other one did.
I don't think it's any bigger than that last one.
Yeah. Oh, yeah.
I think it's, yeah.
Well,
Same size.
Can you pan all the way over?
fingers crossed.
Is that all the way?
Where's this one going?
Next jar. 3.
The last time we put all of her sea pens in the same jar because I think she was just going to group them all as one sample, so she had some repetition within a site.
Okay.
So I wouldn't be surprised. I think we should keep this in the same
Okay.
jar.
I won't make a new sample number then. I'll just count this as part of the same as the last.
Yes.
Yep.
Yes. Thanks. 2 sea pigs.
So, Steve, no target needed then here?
There's no target needed here.
Roger.
I think we're close enough.
Yeah, I would just want to make sure she has enough statistical power to draw some kind of conclusion. And if one isn't enough, I wouldn't want it to go to waste either.
I just grabbed the hose.
Yeah.
Thing's just not worth the trouble. Need to
Yeah.
straighten the arm. Yeah.
Yeah.
So we're going jar 2. Yep.
Can we actually zoom out so I can see the arm? Or Al, can we zoom out a little bit? Yeah.
You can just rotate the jaw a little bit.
Yeah.
Other way.
Oh.
I'm looking at my meters. You guys are incredibly quiet over there. Can barely hear you. And my meters can't hear you either. So it's not me.
Your meters.
It's not me.
No, Paul too. We couldn't hear him. I can always hear Paul.
Oh, you ready for juice?
Are we on it? Hold on 1 sec.
Something tells me this is going to be funny.
You ready?
Yep.
First you beat it over the head. Now you're going to...
Oh, you're...
Oh, that it's... But... But thing. Oh, we got to get the bucket up there. Shit.
Here comes the pig.
Yeah, there's the dirt. This is our
Aw.
magic trick. When the dirt clears, there will be 2.
Yeah.
Sorry?
I think you got 2 in there.
You wanna...
Yep, you got 2.
That looks like 2 piggies. Thank you very kindly. And then I think we can also take a Niskin here as well.
All right.
Since we haven't done any of those yet.
You wanna
I think it might
hose
be.
abuse? Hose abuse.
I think
A separate target for the Niskin?
Let's let go of that thing.
yeah.
No, not necessarily.
No.
I think this is all generally the same area.
I was thinking if
Okay, so Niskin too.
Yeah.
if you think we can budget enough bottles, we have 6 total. I think we should take 2 here just so we have enough water for all the different parties who were interested in having water filtered. I don't know if Meredith wants a liter here, but if we take 2, we can at least accommodate microplastics, the POM, and the eDNA.
Yeah, the eDNA could probably be either used as a blank or used for the C-pens. So I'll take a note of what we found in the area when we pulled it.
Okay.
So it looks like 2 are fired already? Is that right?
No, none have been
None?
fired yet.
Oh, I can't.
We were just discussing whether or not to take two bottles in this one sample.
Okay. So you're going for 1 then.
This will be 064.
665.
Get away from my camera with your nasty old arm.
Steve, what was 64?
So push cores are discrete. So push core 1 and push core 2.
Okay, thanks.
6.5 is the Niskin.
Roger.
Yeah, we're gonna take 2 bottles here, 1 and 2.
And we decided not a separate target. It'll be same target as everything else.
Yeah. Copy that.
And each Niskin is separate sample number, Steve? Or same?
No, it's gonna be just linked.
Roger.
So just all under 6.5.
So Niskin 1 and 2 will be 065.
Yeah, we're just treating it as 10 liters, basically.
Got it. Okay.
Did it go?
I think it already went. Don't yank on that. Yeah, I think it already went. Yeah, don't keep yanking on them, you break them.
Yeah.
Up.
Wait.
Yeah.
Did it go?
2's vertical.
Al's got a better view over there if he wants.
Yeah, with the monitor right up in my face.
Okay, we've got 9 minutes left to watch.
All right. Did some excellent sampling. And unless anyone sees... Oh, yeah.
I feel redundant.
Okay, is that good? Yep.
We've got Meredith now in the science chat as she's preparing to take over a midnight to 4 a.m. watch. I believe with Lisa, so we're also waiting to see Lisa in the chat as well.
Get a jump zoom there, Al?
Any zooms of anything you want to take here, Science, in the last couple of minutes?
There was a purplish anemone, purpley blue, which I don't think we'd been seeing a lot of before. I don't think we have a zoom on yet.
Where is that at?
Might have been on the way to get the...
Yeah, when we were looking for the red radicipes, and there was this kind of big piece of kelp on the ground. It's been pretty interesting just to see a different set of muddy bottom inverts because I've never been down to this depth before.
I completely agree, Abby.
Mm.
It's been a little less rugose, but there's been a lot of creatures we don't normally see, for example, in the Channel Island Sanctuary.
Yeah, the sea pigs and the strange hydroids.
Where was that seaweed at? Remember?
No, there's something kind of purple down there to the bottom left. I don't think that was it, though. Is that another octopus?
Whoa, where?
Oh.
All right. There it goes. Nope, 2 sea cucumbers.
Oh, was that a bubble
Oh.
of oil that just went by?
A what?
A bubble of oil. I don't think there's no octopus. They're very dirty.
No, going backwards. Roger.
Hmm.
Seen anything tempting here? What are we looking for? A purple urchin?
I think they were anemones.
Bob, do you normally have that flicker and bubble cam right in the middle?
Do I want it?
No, do you normally have that?
Oh, I don't want it.
Okay, so it's normally there.
Bob, you want a junk suit?
Doesn't, sure.
It's a lot of junk.
Have we had a zoom on any of these crabs yet?
Oh, where was the crab?
What? There's a
Can you
white
zoom
one
out?
to the
Can you
right.
zoom out? Yeah, over there. Oh, that's what's wrong. Yeah, because we were doing the Niskin. Sorry about that. Oop. Hang on. Diddling the wrong knobs here. I accidentally rotated the wrong knob. I had to get back to normal.
It's a wee crab, Bob. We need to get close.
And I get close. Up close and personal with a crab.
Or is it Menidiopsis, a little squat lobster?
All right, you want to zoom in on him?
Oh, that was a pretty good pan and tilt while I was zooming. I almost nailed it.
Cool.
And I'm out.
Nice zoom.
Alrighty, Shoreside, we are starting our watch change here. Have a lovely evening. We'll see you tomorrow.
You too. Thank you.
Thanks, everybody. Great watch.
Have a good night.
So while they're doing that, just really quickly before Guy had gotten off, we're just past 5B III, and I think we all agreed that 5C and 5D will also likely be mud because they look like they're kind of within that canyon, which has been muddy bottom. And so we were just going to shoot straight for 5E and kind of follow up the nose of the canyon to 5E and then start moving southeast. I'll wait for Lisa and or Meredith to hop on just to make sure we're all on the same page before I get off. Good morning. We've got your radicipes we just collected. You're pretty cool. Yeah, you sound a little quiet. Do you agree, Meredith, or is that just me? Yeah. We got two. Actually hoping Taylor Ann can provide some clarification. We're still in the same area if she needs or wants another, but we got two. Since her instructions in the sample protocol said 1 to 2 sea pens, so we got 2 sea pigs, just to make sure. And then we got 2 sediment cores, and we also fired 2 Niskins here as well. Yeah, that was a sea wet. Okay. Yeah, guys take, and Kurt supported it too, was just to go from 5B III to 5E to hopefully stay on this upper ridge and hope for some hard habitat before heading north, southeast. Mm-hmm. Absolutely. Do you want me to stay on for that, or do you have it? Yes. Yeah. Yeah. It's pretty simple.
All right. How are we doing up here? Getting
Oh.
set up?
Thanks, Lizzie.
All right. Good night. Have a great dive. Fingers crossed for some hard habitat.
Hello, scientists ashore. This is Rennie in the nav seat. Right now we are held position here, and as I understand it, we are headed towards 5E, skipping 5C and D.
Roger that. Yes.
Okay. So, I can just head in that general direction, and we'll just kind of keep a pace of not really stopping. We can stop to look at stuff, but I won't really stop the boat. And then if we do need to stop the ship, it will to collect something. Unless it's quick, it'll probably be 20 minutes, half an hour, something like that.
Okay, so just so that I'm clear, if we're going to collect something, it should be within a window of 20 to 30 minutes because of the lag.
Yeah, we'll be quite a bit laid back, I think. And so in order to stop Argus and come back to where the site is, that'll probably take, yeah, I'd say 20 to half an hour, and then there's the collection and then moving back forward. It's been taking about, from here, when I start moving the ship, it'll probably be about 10 minutes before Argus moves.
Okay.
So that's just like in the back of our heads. We can of course stop at any time, but that's the
right. But realize
penalty.
if we're going to collect, if we're going to collect, it should happen within a 20-minute window. Nothing complicated.
Oh no, we can like once we stop, we have to just come back and collect. It'll take that long to come back to actually get it.
We have about probably a 60 to 90 second window. We'll see. We can test it.
Yeah, we'll see how it goes.
Thanks, Kim. I was trying to sort of understand what we could do. So we should just keep on moving.
Yeah, and if we see something spectacular to sample, we'll absolutely stop. It'll just take a bit of time.
Okay. I got it.
Argus is a giant pendulum, and we have to go where it goes. So it drags behind the ship, and then when we stop the ship, it keeps swinging in, as we call it.
Yeah, it keeps.
Or as Rennie calls it, the layback. So it swings in at kind of a constant speed.
Okay,
we'll just observe.
I'm going to start the ship move now, unless we have anything else to look at here. Which way am I going, huh? Let's go.
Where are you going?
050.
050.
So Meredith, did you understand?
Oh, sorry. It looks like they stepped back for the last sample, Rennie. Is that true?
Yep, they did.
How far did they step back?
Looks like there may have been a heading change in there too. Hard to say, but it looks 50 meters, something like that. Yeah, 50 meters back. Okay, on our way. Bridge nav.
Sorry, go ahead. Didn't mean to cut you off for sure there.
Oh, no. You go first. It's, we wouldn't be here without your navigation and piloting. So I'd ask Meredith, because there was a specimen right there, that the long, white, sort of snake-like undulating tails that we're seeing, are those a worm?
So originally, early in the dive, I had ID'd those as an acorn worm, possibly. I was, just looking around at some stuff online, but I actually just got an email from Erica Frew, who said that Erica Burton looked and says that those are actually a species of hydroid that they've identified from previous dives at Davidson and Sir Ridge. So those are hydroids.
Oh, good. So they are within the they are filter feeders. And what is their name?
Let me pull that up and see if I've got
right. They're within the family. I went back in the chat and I was looking. So they're within the family Candelabridae.
Yes.
Okay, cool. Thank you. How you doing, Meredith? Here we are again.
I'm doing
midnight to 4.
Doing pretty good. Kind of waking up again.
Yep. How is the Nautilus crew doing?
Good. Good.
This fine morning.
Excellent.
Yeah.
So for the folks who might be listening in, should we do a quick round of introductions so they know who's talking? We can go first for once.
Sure.
Okay, good morning, or as I've been saying, deep morning to you all. This is Lisa Vonik. I work for the Office of National Marine Sanctuaries in the West Coast Regional Office, which is situated in Monterey, California, not too far from here. I am talking to you from my home office on the north side of Monterey Bay, Aptos, and I'm the what am I? I am the policy coordinator for the 5 West Coast National Marine Sanctuaries.
This is Meredith Everett. I'm in the other science position. I am also broadcasting from home tonight, or early this morning. I'm up in Seattle, Washington, where I work at the Northwest Fisheries Science Center for NOAA, and I am a geneticist. I work with the Deep Sea Coral Research and Technology Program doing genetics of corals, deep sea corals and sponges, taxonomic identification and some population genetics, and also environmental DNA research on them.
This is Stephen Matter, video engineer in the 12 to 4 watch.
Dan on the winch.
Jake Bonney, I'm in the Hercules right now, normally your Argus pilot.
This is Renato Kane, the navigator on this watch.
And this is Taylor Ann Smith, the data logger.
Okay. So Taylor Ann, you saw the collections that they just made.
I did.
Do you,
yeah.
Yeah. So do you want to tell folks about why they made those collections?
Sure.
Or
So
could you? Thank you.
yeah, I'm interested in what types and concentrations of microplastics are existing in different areas of the deep sea. So I'm trying to look at a vertical profile of the water column and seeing what concentrations and types of microplastics are associated with different water mass properties. But I'm also looking at organisms and some drift kelp. So I've been collecting some urchins, which are detritivores, as well as like some sea pigs and sea pens, which feed in the water column, to see if these different types of feeders are ingesting microplastics, and if so, what kinds and how much. And the kelp is something that's kind of creative that I got interested in due to the other part of my project, which is more near shore. So I'll be looking at microplastics in kelp forests as well because of the different physical properties or physical things that happen in the kelp forest compared to the open ocean. So the thick densities of kelp actually attenuates water flow, which means it just slows down the flow of water due to the thick presence of the kelp. And this allows water to sit in one place for a longer period of time. And so I'm hypothesizing that this could allow for microplastics to accumulate and kind of act as hotspots in kelp forests. And
050.
so I'll be seeing if kelp itself is a vector for microplastics, seeing if their surfaces are mucousy enough to kind of adhere these microplastics. And then potentially, if there are things like drift kelp in the deep sea, this could be carrying along microplastics with them into new areas that they weren't before. But I don't know, that's just a little creative part. I try to look at it to connect my project, because initially I wasn't going to do a deep sea portion, but that's one thing that's been the bright side of COVID. Yeah, COVID kind of brought that upon me because I wasn't sure if I was going to be able to do my original thesis project. And I was coming out here as a science manager in training and just was like, hey, I have these ideas. Is there any way I could, collect some samples and get something for my thesis in case I don't have a project anymore? And yeah, so that's how I'm out here doing a little research. So it's cool.
Well, you're doing research and you're really helping us with
yeah,
doing the data logging. So
it's great.
That's been great.
Get to do science on all the sides. Yeah, it's nice to be able to see all these samples when they come up and learn about them and see them up close. And yeah, it's been a great experience. I loved it last year, so I was really, really thankful to come back again this year.
So those little brown things are sea cucumbers? Or are they're not feather boa. Those are just.
I think those are another species of sea cucumber, yeah.
Oh yeah, okay. Just curious. I liked how I heard Lizzie at one point, because we've been seeing them all along. This dive started at 4 p.m. yesterday, so we're already 8 hours into the dive. That they all seem to be evenly dispersed somewhat, almost as if they have their territories.
All in their own little patch.
Yes, their own little patch of slurping up this mud and sifting through it, right? And then they leave behind these very neat poop packages.
Mm-hmm.
They're
So we've got a
coiled.
pycnogonid that just went under the lasers.
Yes. So it's a spider crab.
It's a sea spider. They're actually
Yes.
more closely related to the arachnids than the other decapods.
So chat, I saw also in the science chat that Chad King, who is a scientist from the Monterey Bay National Marine Sanctuary, and he's the chief scientist on the Nautilus cruise portion that visited Davidson Seamount. And he was saying that for these sea pigs, the scotoplanes, that we should occasionally flip one over to see if there is a crab, a juvenile crab that is sort of hitching a ride and under the belly of the sea pig, and they are hiding from potentially this is an adaptation to hide from predation. I thought that was pretty cool when I heard about that. Just like, I guess the sea pig doesn't really have any appendages or a way to shake them off. So maybe they just endure it, or maybe it is truly mutual symbiosis or something.
Yeah, they might not have much of a choice.
Yeah, just sort of like, well, grin and bear it.
There's always a question of in the stuff that's, we rarely have time, or, we're getting a snapshot. It's sometimes difficult to understand if it's a symbiotic relationship or a parasitic relationship.
Yes, that's true.
In his description, he made it sound almost like the crab was carrying the sea pig around, like a little umbrella.
Yeah, yeah.
That's a good way of looking at
it. Yeah, yeah. So it could be that the sea pigs get to a different location more quickly.
Mm-hmm.
Yeah, they get to new food faster than they might otherwise.
Yeah. Yeah.
What's that? I think you're muted.
Yeah, there's just so much of the deep sea, this deep, that we don't really know. It's hard to get here. When you're here, I think, as you just said, Rennie, it's like a snapshot in time. And but with technological advances, and the technology that we're using right now, using these remotely operated vehicles, we are slowly filling in the puzzle pieces.
All right, Roger, coming down.